to the next bit. So an actual sort of comparison between mitosis and meiosis, very common question. Whenever you've got two similar processes in biology, you just know there's going to be a compare and contrast question in there. So it's important that we can, and whenever you are making any kind of compare and contrast questions, it's really good to be able to separate out what aspects of them you're comparing. So with respect to how many divisions there are. It's one cell division and makes two daughter cells. Two cell divisions makes four daughter cells. The next aspect is whether there is recombination. What do we mean here by recombination? We mean recombining DNA sources that are not identical. Daughter cells end up being genetically identical. So you can see here that they're identical. They contain the same copy number of chromosomes and an equal mix of maternal versus paternal chromosomes. Whereas little cells are haploid and they only have one set of chromosomes. Bear in mind that this idea of haploid versus diploid, as we saw in that question, can also be somatic versus germ cells or gamete cells. Not germ cells, gamete cells. Let's have a look at an example of one of these extended response questions comparing mitosis and meiosis. I'm going to give you a few minutes to try and come up with the answer yourself before we look at the answer together. But let's first do our strategy of breaking down the question. Explain how meiosis increases genetic variation and compare the products of meiosis and mitosis. This is actually a pretty generous phrasing, I would say, because sometimes they might literally just say compare and contrast meiosis and mitosis. Whereas here we're kind of like told that you also need to make sure to include in your answer the products of mitosis and meiosis. We would do so anyway, even if we weren't told, because that's part of what comparing mitosis and meiosis is. But now we're told explicitly. 1 mark is given for the quality of written communication. So again, what is my context? It's this first bit here. It's, sorry. So sorry. I was so used to, see, my bad. I jumped ahead. My context here is that I am dealing with mitosis and meiosis. So those are the divisions I'm going to compare. My command word is explain. And explain with some detail as well, since it's an extended response question. And finally, my direction is that since I'm comparing, let me just change color here. Since I am comparing the products, I need to include comparative language, comparative adjectives or language. While, whereas, things like that. Oh, sorry, I did it again. So sorry. Take about sort of 3 minutes to 4 minutes to have a go at the answer. Okay. Let's have a look at the breakdown. So out of those 3 marks, so 3 will go towards explaining how meiosis contributes to genetic variation. About 2 of the marks are going to contrast the products of each one of mitosis and meiosis. And then finally, of course, 1 mark goes for the quality of the QWC. Sometimes these are abbreviated in the mark scheme for examiners. Quality of written communication. So let's see the answer together. Mitosis produces. So we're starting by comparing the products. Mitosis produces 2 genetically identical diploid daughter cells. So genetically identical, let me just use red. Genetically identical diploid daughter cells, that's 1 mark because those are the products. While meiosis produces 4 genetically distinct haploid daughter cells, that's my 2nd mark. So those are my 2 marks for comparing the products. Variation meiosis can be caused by recombination between homologous chromosomes, i.e. crossing over during prophase, that's 1 mark. You get 1 mark for stating that it's between homologous chromosomes, and you get another mark for stating where, when, and where it happens during prophase 1. Or by the independent assortment of homologous chromosomes, which occurs both in metaphase 1, so independent assortment of homologous chromosomes is 1 mark, which occurs in metaphase 1, so homologous chromosomes in metaphase 1 and sister chromatids in metaphase 2. It's really important that you distinguish. Don't just say homologous chromosomes and sister chromatids. Define which cell division they take place in. And that's another mark here.